Hello everybody and welcome to today's webinar, Overcoming Manufacturing Challenges for Gene-Enabled Cell Therapies, hosted in partnership with Catalent. I am Georgie Makin, the editor at Facilitate and moderator for today's discussion. Today I'm joined by a great panel who will shortly be introduced to you. But before we get started, I just wanted to take a second to run through a little bit of housekeeping. First of all, I would just like to remind you that we encourage our audience to submit questions to our panelists. So please do submit your questions via the Q&A widget accessible via the bottom of your screen. We may also attempt to answer your questions offline should we run out of time. This session is being recorded and will be available on demand shortly after the conclusion of the event. Details for on-demand viewing will be sent to you once the recording is available. Now, before I introduce the panelists for today's session, I would like to hand over to Phil McCauley from Catalent for a brief introduction. Over to you, Phil.
Thanks, Georgie. Hello, everyone, and thank you for joining today's webinar. Catalent is proud to sponsor this event on overcoming manufacturing challenges for gene-enabled cell therapies. Today's topic is very significant to Catalent as it supports our broader mission of making more treatments accessible to patients. The cell therapy industry has seen aggressive growth, in part due to market approvals for autologous CAR-T therapies, which offer durable cures to patients suffering from blood cancers. The lessons learned from these first-generation CAR-T therapies have led to engineering modifications for the next generation of therapies, such as allogeneic modalities and application for broader immune cell types. More cell therapies are making progress towards late-stage manufacturing, some under the Fast Track designation, demonstrating promising outcomes in oncology, regenerative medicine, and other diseases. Both cell therapy innovators and manufacturing organizations are employing novel technologies and methodologies to adapt to an accelerated and evolving clinical landscape. As there are still a high number of therapies in preclinical phase, there will be a heavy push to move these drugs to commercial readiness while managing potential financial burden. Major factors like supply chain complexity, process efficiency, and a solid understanding of regulatory requirements around handling human source material are critical to the success of these medicines. Coupled with supply chain complexity is the inherent challenge that each cell type has its unique biological blueprint, making it difficult to move towards a standard manufacturing system for scalability. Even for gene therapies, although AAV vectors are leading the way, there are multiple options for delivery, including lentivirus, retroviruses, and other that require custom optimization. As a manufacturing partner, we are staying at the forefront of these market shifts and flexing our capabilities and capacity to meet the growing demand. Efforts are underway with our teams to drive process efficiencies at scale for these next generation modalities. Additionally, as more products progress to commercial approvals, we are making significant investments to add new manufacturing capacity to meet the aggressive demands for these promising cures. To briefly summarize our capabilities, Catalent Cell and Gene Therapy offers fully integrated solutions for manufacturing CAR-T immunotherapies and beyond. Our capabilities range from plasmid DNA manufacturing, viral vector production, to autologous and allogeneic cell therapy manufacturing. We have a global cell and gene therapy network of clinical and commercial manufacturing facilities and fill-finish capabilities across the US and Europe. I'm looking forward to an engaging discussion from the panel about the progress in leveraging manufacturing expertise to overcome key challenges attributed to commercial scale cell therapies. I'll hand it back over to Georgie, our moderator.
Thank you, Phil, for that introduction. I'd now like to give our panelists the opportunity to briefly introduce themselves. So Kelly, would you like to start?
Thank you, Georgie. So I'm Kelly Davis-Clays. I'm the Director of Global Regulatory Affairs here at Catalent. I've been with Catalent for 2.5 years. It's been an exciting time to get to understand and learn all about the support services that we can provide here. Prior to joining Catalent, I was part of a large pharma company in which we manufactured both traditional and biologics as well as medical devices. All of my experience has been either in quality or regulatory and has again been across the globe.
Excellent, thank you. And Tim, would you like to introduce yourself?
Yes, thank you, Georgie. I'm Tim Ferris. My background is as a developer, originally in gene therapies and cell therapies. But since 2013, I've been in consulting. And me and my team, we consult on gene therapies, cell therapies. We include a lot of work on stem cells, genome editing, viral vectors, all this field, really. My screen showed me as Chief Scientific Officer of ERA, which I was, but we have now been incorporated into the Biopharma Excellence brand enterprise, which is part of PharmaLex, and is building up a bigger team of expertise across the cell and gene therapy space. So very much looking forward to this.
Excellent, thank you. And Knut?
Yeah, thank you, Georgie. So I'm Knut Nis, I'm the Chief Technology Officer for Mustang Bio. Mustang is a company focusing on autologous therapies, a few CAR-Ts, and a gene therapy for XKID. Prior to Mustang, I was actually working at Biogen on the zinc finger technology for sickle cell and beta cells, so I gained some experience there. And prior to that, I was with Novartis working on the CAR-19, that's like Kymriah. And then way back, I was at Pfizer Regenerative Medicine when cell therapies were all about adult stem cells. And I was heading up the adult stem cell space at that time. So thank you for the opportunity to participate.
That's no problem. Thank you all for joining us today. To kickstart today's session, I'd just like to start by asking, what are the critical manufacturing challenges associated with allogeneic gene-enabled cell therapies as they mature to commercial scale? Kelly, could you start by describing some of those challenges associated particularly with GMP?
Sure, I would love to. I think that as we think about cell therapy, the one thing that comes to mind immediately is how fast-paced this area is. We go from preclinical to all through the clinical phases to commercial readiness in lightning speed, if you compare it to other areas of the pharmaceutical and biologics world. So I think the biggest hurdle that we have to, or challenges that we have to overcome, is making sure that our validations, our qualifications keep up. Obviously, we're not going to go as quickly, or we're not going to be at the same pace as you would in a traditional pharma or biologics area, because there's much more time. So I think it's very key that as you're going from clinical and you're getting closer to that commercial state, that you look very closely at your qualifications and validations, whether that is your manufacturing processes or it is your analytical test methods, because obviously I'm looking at this from a regulatory affairs perspective. Whether you're a cell therapy, gene therapy, biologics, traditional pharmaceutical product, you still have to have the same GMPs when you get to commercial. And a regulatory submission still needs to be able to prove to the agency that your product is safe, it's effective, you can manufacture it consistently. So I think looking at those qualifications and validations is very key and critical. Another area that I see often is that some of our partners, their supply chain is not as robust. So I would say also make sure that the supply chain that you start with in clinical can either transition to be able to provide GMP material, or you have an alternate source that you are identifying as you progress through the clinical trials. I would say those are the biggest challenges that I see in cell therapy.
Thank you. And Tim, I know we discussed offline a bit more about comparability, design and analytics. Could you speak a bit more to those key areas?
Yes, it's one of the things that clients come to us most often with at the moment, is problems getting regulatory acceptability, manufacturing changes, and it's a huge issue in this field. And making changes to the manufacturing, it's inevitable, but it requires a lot of planning, because it's very difficult sometimes to assure that the product isn't changed, to understand the product sufficiently, to understand the analytics, to understand the critical quality attributes, to make the changes at the right time. So many companies want to make big changes in pivotal clinical studies, and then some of these products are actually very sensitive to manufacturing process changes, especially say products that include stem cells or derived from stem cells. Small manufacturing changes can make a big difference. And then the analytics is not always up to scratch when it comes to assuring regulators that nothing has changed, certainly nothing critical. So this is a very big issue for the field.
And Knut, what would your perspective be on some of those key manufacturing challenges? Are there any that we've missed or that you think we should be highlighting?
No, not missed. I would say, like, I would comment on both. So first of all, the comparability, I absolutely agree. Comparability, sometimes I see people taking that too lightly and make changes late in the processing, even in pivotal trials. So my advice has always been, no, don't make changes in pivotal, make them before. So the philosophy we are adhering to is basically our first patient in phase 1 should look like the first patient in commercial from a processing point of view. And I would always argue that's the most safe approach, because one thing to remember about comparability is there's no clear checkbox exercise where you say, okay, if I show this, and this, I have comparability. The agency will look at the big picture and all the data and decide by themselves. And what your opinion is in that regard is really not meaningful, so to speak. They will decide if you're comparable or not. So, and then the second part on comparability that I think coming to light now a little bit more is also comparability is not just the cell processing, it's also the drug substance, in most cases, the lentivirus. So when you change the source of the lentivirus, you need to have comparability there. Again, an exercise that can be fairly complicated, and you need to be prepared for that early on. And then lastly, comment on the supply chain. I think what we see now, especially nowadays with all these supply chain issues, is, originally it was great. There are different levels of material. You have material that were made according to GMP. You have material, we can use material that is USB or, injectable grade, like saline, for example, or protamine sulfate in our case. You strive to get to the highest level of safety. So if there's a USB material, an injectable grade material available, you go with that. Turns out that's probably not a good choice, because when there is a product shortage, you as a manufacturer will always be on the bottom of a list who gets material. So sometimes the agency pushes us to go to these kind of injectable grade material. In our case, as I mentioned, an example from our side is protamine sulfate. There's a shortage. We have a really hard time getting it. There's plenty of material made according to GMP. So would we have that, we wouldn't have the problem. So I think supply chain is something to be taken very serious early on as well.
So apologies. I think there you've highlighted a little bit about my next question, really, about getting in there early. That to me sounds like a bit of an obvious solution. So are there any other obvious solutions to those challenges that we've discussed? Tim, do you want to start?
Well, yes, one is to understand the product from the start, because until you understand the product, you don't know what's really going to make a big difference to it. And there's so much talk about potency, but one of the ones that people often forget that really does apply from the very start is what you're going to dose on. You need to understand what is making your product tick, which is not always very straightforward for a complex cell therapy. There may be more than one component in there, and there may be other components that you discount. And this understanding of the product is absolutely critical to that. And the other aspect is going to be what makes it safe as well, which is often very much neglected from the start.
And Kelly?
I would say from a regulatory perspective, don't forget the basics of regulatory, right? So at the end of the day, this is some new technology, it's advancing therapies, but there are basic regulatory expectations that the agency is going to have that you need to keep a focus on. So getting back to the basics of the regulatory requirements. I would also recommend that I recommend for all of our clients that they speak to the agency. There's nothing wrong with reaching out to your reviewer or your division and asking for a meeting so that you can have some of those conversations up front. It can be very beneficial to get their feedback and be able to help you model and plan your regulatory strategy.
And Knut, do you have any obvious solutions to some of those challenges we've raised?
Yeah, solutions, I don't know, but I agree. Certainly, like talking to the agency as early as you can is always a good idea. My experience has been that they're very helpful and they want you to succeed. So they give you usually the advice you need, even if you don't like it. But we're entering a phase now within the space where we have more and more assets entering pivotal trial, late stage trials, and so on. So the questions become new, like something like a potency assay that hasn't been really on the radar for a long time is now becoming more and more important to many people. And I think that's where we need to work with the agencies, not just here in the US in general, about educating them around them. So if you look in the space, potency assay, for example, is one that is often a CMC hold item for late stage trials.
So I think we've touched on regulatory quite a bit there, and moving on to our next segment, that's certainly going to be a bit of a key feature, but I just wanted to take a second to remind the audience that we are accepting audience submitted questions. So if you do have any questions for our panel, make sure you send them in using the Q&A widget. So moving on to that next segment, how has the regulatory landscape evolved for cell therapies in the past few years? And what is expected, I suppose, in the coming years that innovators should be paying attention to? Kelly, I don't know if you'd like to start with this one.
Sure. So I think that, with any new areas that we've seen with the agencies, I guess just to start, the agencies typically begin very basically. They provide very basic guidances and regulations, and as they experience and learn the technologies, their guidances and regulations become more and more specific. And I think that you're starting to see this with the cell therapy area as well. We have some very general guidance documents that are specific to cell therapy. Again, that's just another reason why you should talk to the agency, is because you need to have those meetings to fill in the gaps, so to speak. But I think as we look in the future, we'll see more and more guidance documents be published so that you can get more specific documents out there for general guidance. Never negates having that conversation with the agency, but I think as you watch the regulatory landscape, you're seeing that the guidances are getting more specific, giving much more details of things that you can use to plan your regulatory strategy. Now, of course, we have a ways to go. This is still somewhat new, at least in a regulatory time frame, so to speak. As I said, the agency moves a bit slow, but I would definitely say that we can see some evolution that's starting, and I would just expect to get more and more agency input in more formal types of communication, like a guidance document.
With these guidance documents evolving, does that mean that we might be moving to an environment where maybe we no longer have to reinvent, or we may not have to reinvent the wheel as such? There might be workflows that you can just adopt almost immediately. Tim, I don't know if you want to take that one.
Well, I like to think, and I think it's certainly true, that actually the forefront of product development is always going to be ahead of the guidelines. Regulators are great, but the innovators are more innovative, and so there's always that forefront. And I don't think we are looking at a field that will be, and in fact, it'd be less interesting if it was true, that it will be fully covered by the guidelines. Mm-hmm. Having said that, the guidelines are making progress, and they are actually adapting. I think one very big way they're adapting is the early products, a lot of autologous products, fairly exceptional cases, many highly variable. We're now, I think there's much more focus on more consistent allogeneic products where regulators are saying, okay, this really ought to be what it says on the tin. Mm-hmm. It needs to match the label. There ought to be much more consistency in the product. These are some of the parameters we know we want, and they understand a lot of those and are starting to, I would say, impose them more, rather than waiting to be told what they might be able to get.
Thank you. And Knut, you've worked on quite a few different advanced therapeutics over the years. Yeah. What has your experience been in working with regulatory agencies as you're moving them through the pipeline?
Yeah, I would say I hate to age myself, but over the last 10 years, I see a shift from, in the early days, it was like they were as confused as we were in terms of what is possible, what needs to be done. I think the agencies are much more mature now in understanding how cell and gene therapy works, but they also live, of course, in a different environment where things move a little slower. So, I mentioned earlier the potency assay. The current guideline on the potency assay is 10 years old, which by itself doesn't sound like a big problem. But if you remember, 10 years ago, cell therapies were allogeneic MSCs. There was little beyond that. And today, I would argue that the average cell therapy is an autologous T cell. So, there are guidelines that are in desperate need of updating. There are guidelines that, are still valid, and there are guidelines that would still need to develop going forward, especially now with the gene editing. I think we don't understand necessarily the gene editing space, the regulatory environment that well. So, for example, if you look at the recent Allogen example with the chromosomal aberration, I think it's going to be very interesting to see how they're going to solve that problem because there's really no good solution that I can see. So I think, it continues to be a journey where regulators and us are working hand in hand, but at the same time, who's pulling who is still remains to be seen.
And on that theme, how might the communication, interaction, or I suppose relationship change with different regulatory agencies to reflect shifting focuses of cell therapy products? Kelly, I don't know if you've got, if you want to start with this one.
Sure. So I think, as Knut mentioned, is we have guidance documents, and as I mentioned before, the agencies move very, very slow to change those regulations. I think it is definitely we need to take an opportunity to interact with the agency. This in cell therapy, it's not like traditional pharmaceuticals or biologics where this is well known. The agency is well trained and understanding of the general principles. In this manner, we almost need to educate the FDA. So this is where we have to tell them, and where Knut mentioned too, is we're starting to see some shifts, but it's almost where we're telling them this is the capabilities that we have. How do you interact and get that interaction? It's really with communication. You have to interact with the agencies and help them understand your product. They'll let you know if what you are presenting they don't feel comfortable with, or they think from a commercial launch perspective there are some issues. But I really think it is very open communications. That's a very key piece.
And Tim, do you have any insight you'd like to share on this one?
Well, yes, I think the agency, actually, I'm worried that the agencies, particularly the FDA, is getting overwhelmed with so much development, so many products coming towards them. And we've seen this from the FDA in particular for extending the timelines they can do for meetings. And both the FDA and European agencies are having more and more written answers only, but then some of that's because of COVID. But also the time and the recent announcement from the FDA that they're thinking maybe they're going to manage less for one-to-one meetings and make more generic pronouncements or guidances. I think it's going to be, the field really needs these one-to-one interactions, and we've had some great ones. The Interact meetings, they've really been superb ones we've had recently. But I think we're going to find that the agencies just don't have the capacity, and I think that's going to be quite challenging, especially with other difficulties in finding the right expertise outside of agencies as well. Relatively few agencies that one really wants to go to for the most cutting-edge projects.
Excellent, thank you. I'm going to take a quick pause from the segment that we're on at the moment to address an audience question. So just a quick reminder to our audience, please do send them in and we will try and address as many as we can during this session. So this question asks, with many new cell and gene therapies coming through the system, in addition to the supply chain constraints as mentioned, are we seeing manufacturing capacity constraints arise in the US? Knut, I don't know if you'd like to take that one.
Yeah, I would definitely agree. I think we see that. There's a reason Mustang, for example, built its own manufacturing capacity facility, and the reason was that we foresaw that it's going to be really hard to bring 4 or 5 CAR Ts and a gene therapy to other places in the current world. Now, the good news is, now with the cell and gene therapy space thriving and frankly, investor money flowing into it, there are new entities coming along, new CDMOs that are built. Existing CDMOs build out their capacity. So there's more capacity build in general. And in addition to companies building their own capacities. But at this point, I would say in my view, there's a constraint. The supply chain issue is making things a little bit worse in the sense of, that's a second arm of problems we have. We can't manufacture a product if we don't have the materials. So, hopefully that will be supply chain issues at least will be resolved. And hopefully we will see more growth and expansion of existing space. But at this point, I would agree, it's a problem. Oh, sorry, you're on mute.
Apologies. Thank you for that one. I want to return back to our main middle segment here. So we were talking a lot about regulatory or agency associated challenges in quite a generic way there, really. But are there any differences between agencies in the US, Europe, or worldwide that perhaps should be highlighted from your experience? Tim, I don't know if you'd like to start with this one.
Well, there are, but they can easily be exaggerated. A good product in one jurisdiction is mostly a good product in another, but there are certain things you want to watch out for. Sure, the FDA is perhaps leading the way on many things, especially like genomic therapies, off-target analysis, the greater depth of that. We see much more foresight, I think, from the FDA than we do from European agencies in this. But there are a few things to be careful of. I mean, if you look at Europe, I would say there are about 3 things to be very careful about. One is sometimes the donor requirements can vary across jurisdiction for allogeneic cells. Number 2, very important, the Europeans have been very strict on GMP requirements for key starting materials. Although, to be honest, I think the FDA is pretty much matching that now. And another one that causes developers quite a lot of problems frequently is if they think they can rely on US DMFs too long. US file DMFs don't work for filings for marketing authorization in Europe. And alternative solutions must be found, and that can be quite challenging.
Excellent, thank you. Does anyone else have anything to add about any differences between agencies that you might have experienced?
I think that, as Tim mentioned at the very end, when you have a supplier who has a US drug master file that's filed with the US, a lot of times that is proprietary information. And so as you're expanding out into other regions, you really do need to understand how that information is going to be supplied to you, or if they won't supply it to you, and how you're going to circumvent that potentially with other suppliers. So I think from a regulatory perspective, that's always key, because if that information isn't available to go into your submission, then it stops that path into that country.
Excellent, thank you. I think I'd also like to now move on to our final segment, talking more about innovators. We've been talking a lot about innovators and progressing along the development pathways. So if we are focusing on innovators, what is it that you think they need to know or understand when developing these products as we've described? Tim, yeah, if you'd like to start with this one.
Well, one thing I used to advise clients on, I still do actually, but it used to be a bit of a speciality, was designing gene therapy vectors, things not to do in some ways, and how to get that design right. And it also applies to certain cell lines. But with vectors, sometimes what you start with is very hard to change. So you need to get everything right from that perspective. So if you're an innovator, this is one element of product design that really needs to come in early. There's the other aspect, I think, that one wants to emphasize at this stage is it's very dangerous to start a product development until you know your process for manufacturing it is robust and is going to consistently produce the same product. Just because you can do it once or twice and can do it in the lab, if this is then going to start to produce different product later on, you've got big problems. So that's the second point I would leave you with.
Excellent, thank you. And how about, oh, sorry, apologies. And Kelly, is there anything you'd like to add to that? Just returning to that focus on innovators.
Yeah, so I would say that I'm going to speak a little selfishly coming from regulatory affairs. Innovators are great from the science perspective. They understand all the nuances and intricacies of their product. A lot of times the innovators that we work with are very passionate about their product and the therapy areas that they're going into. They're very excited about getting this product to the market. They have the patient first in mind. And the one thing I would remind an innovator or a small company is to take a moment to bring regulatory on board, because if you don't have all of your documentation, all of your, and I've said this, I'm repeating myself multiple times, you need your product to be validated, your test methods to be qualified. You have to have this information to be able to show to the agency that your product is reproducible, your impurity levels are acceptable, you have good specifications. As Tim mentioned, it's, you can manufacture your process in produce a very consistent product. I very much strongly always urge our clients to get regulatory involved first, because our job is not just to take all the bits and pieces of the information and write a nice story, so to speak, but it truly is to help get the product approved as quickly as possible. And part of that is to evaluate those documents that you're generating and your clinical trial data that you're generating along the way. We can identify some potential pitfalls during the regulatory review, also provide some insight as you look into after commercialization of how can we set up a submission to make those post-approval changes that might be coming, whether that's site transfer, changes in suppliers, etc. How can we make that a little less painful? So I would, like I said, a little selfishly, strongly encourage a company to get a good, strong regulatory group and consulting team together so that you can look ahead. Otherwise, you may paint yourself into a corner that's very difficult to get out of.
Yeah. So if I may add, and Kelly, you're going to like this, because my thinking is the first two hires you want to make when you start a company is quality and regulatory.
Oh, I do like that, Knut.
And that is really for a specific reason. You, as an innovator, you understand your process, your product, and so on. But often what you don't understand is the regulatory environment you find yourself in when you work in a company. Again, you can't take the learnings from an investigator-led trial that you ran into a commercial IND filing, so to speak. So.
I'd like to stick with you for this next question on that theme. So how would somebody, I suppose if we keep the example of the innovator, how would you then go about acquiring the expertise and the knowledge that you need at that level, and what are the common pitfalls in doing so?
Yeah, so expertise, by the way, is a big issue right now because the field exploded around us, and you see all these, lots of companies being formed over the last few years. But at the same time, there's, a limited talent pool that has experience, right? So when you look at some job descriptions, it makes me laugh because they're asking for things that simply can't exist. 20 years experience, BLA filing in the cell and gene therapy space. So, the expertise, you have to be a little bit more flexible in what you can do. But as I said, before, I think the critical piece here is to be able to think creatively while still being in quality or in the quality mindset, so to speak, or regulatory mindset. So what worked in small molecules and biologics doesn't necessarily work here. So you need to be able to think a little bit outside the box and drive it forward. And then, what's behind that is, a strong process development team that also understands the GMP requirements. So one issue I've seen in the past is, you have a very strong process development team that knows the science of their process and all that, but they don't understand necessarily GMP requirements. And then when you want to move it over to manufacturing, you run into issues. So, again, I think when you build this out, you always want to have GMP quality regulatory as a guide principle.
And Kelly, what would be your opinion here?
I would say definitely start early in looking for that expertise. There's a lot of consulting firms out there. There's a lot of, as Catalent, all of our manufacturing sites are supported by my global regulatory affairs team. We have a wide breadth of regulatory knowledge and experience, and we try to complement our manufacturing expertise with a regulatory guidance. So I think that, look at if you're using a CDMO, look to take that step back and not just be focused on manufacturing and get the product to the clinic or on stability, et cetera, but take a look at what offerings they provide, because in a lot of instances, they can kind of bridge the regulatory. This is what a submission requires, and this is what an analytical test method validation should look like for a support document. But they can talk manufacturing. So they can, a lot of times, walk down the hallway and talk to the PD guy or to the sterility assurance team, et cetera, the quality team, and they can be a good liaison. And that's really at Catalent. What we try to do is we try to walk that middle ground between our clients and our manufacturing sites and try to explain both sides. So I would say, there's a lot of good consulting firms out there, but don't neglect what other options that your CDMO may be able to provide.
And Tim, Knut mentioned about the skills shortage. I wondered if you'd either want to speak a bit more to that or perhaps explain what other things shouldn't be forgotten as we're exploring the idea of expertise and knowledge acquisition or recruitment, I suppose.
Yeah, well, we are, of course, a regulatory consulting firm. And what we do is quite specialist, and it is hard to find the right people. And then again, one of the reasons why we get a lot of business is because the developers also can't find the right people and therefore need people like us. So, I mean, what we do in the regulatory space is we combine not only regulatory, but also with people who've come from product development backgrounds, good scientific backgrounds, who understand the science, understand the manufacturing issues, understand the safety issues as well, which combine with what's often missing from the innovators. Unfortunately, that is a skill set that's in very much short supply. It's what we do, but there really needs to be more of us, to be honest, because it's something that's quite critical. And I think a lot of companies are suffering because they don't easily have access to it or don't know that they need it straight away.
Excellent, thank you. We're going to take another quick pause to address some audience questions, so please continue to submit them as we do have plenty of time. We have one question from, well, I won't say who from actually, but our audience member asks, are there any specific challenges that can be shared when using more than one CMO for producing drug products? They've then gone on to clarify to say that due to time constraints, it's often necessary to use multiple CMOs in at least 3 to 4 regions. So I wondered, if Tim, if you'd like to speak to that.
Yeah, that's terribly challenging in practice, and I can speak to this because when I was a product developer and we had our own manufacturing and outsourced manufacturing, and from those we see, it is so hard, as I alluded to before with compatibility, to get product to be exactly the same out of 2 different manufacturers, let alone 3 or 4. Sometimes it's necessary because maybe they're autologous products that need to be made locally. Rarely I've seen cases where in early stage development you need it for capacity, because this capacity is so hard to find. It's really complicated. What you want to do though is be careful to make sure that the manufacturers you sign up with are going to play ball completely. They are open to full tech transfer, supporting it. They're not hiding too much information or hoping no information. Plus, of course, they're providing all the regulatory support you're going to need. But it's going to be such a challenge that my first advice would be avoid it if you can. And if you have to do it, well, then take those challenges head on, because there's going to be a lot of them.
Does anyone else have any insights for that question there about having multiple CMOs?
Yeah, I mean, I certainly agree with what Tim said, but maybe on the bright side, I see the start of what I would call a global network of CDMOs, right? Like where now the CDMO in Europe is either acquired or partnered with the CDMO in the US. So if you work with these kind of partners, you at least have only one entity to deal with. Still, all the problems that Tim mentioned, of course, exist. Cannot be taken lightly. I agree, Tim, like if you can avoid it, avoid it.
And one thing I didn't mention is analytics. Transferring analytics between different sites adds to the challenge. And if you can't measure, say, the dose equally between sites or the in-process controls, you've got big problems.
Right.
And I think it's not just important and critical things to think about that Tim has mentioned, isn't just to add a second manufacturing site, but it's if you have to transition or do a technical transfer in the process of your clinical trials too. So if you start with one manufacturing site and then you move to another company, those same pitfalls and hurdles and challenges are there as well. And it definitely takes a lot of thinking and preparation. It is not an easy move.
Excellent, thank you. So moving on with the main discussion, we are returning back to a slight regulatory theme, but how are regulatory priorities shifting as cell therapies move from earlier to later stage? Kelly, I don't know if you'd like to start this one.
Sure. So I think that as we're transitioning into a bit more of an established cell therapy system and program, as Knut mentioned earlier, is that it used to be a little bit us leading the agency. And I think that we see shifting regulatory expectations is that there's a more of a foundational understanding. So there are now expectations that the agency has versus us leading them. So I think you're starting to see that transition. We've seen that before as new therapies or technologies have been developed. It has played out, especially here in the US, in a very similar fashion. So I think that you need to make sure that as you are shifting from early to late stage clinicals and into your commercialization, that you, again, I seem to be repeating myself because it's a very important aspect, is understand your process, understand your parameters, know what is critical, know how long where you can shift and maneuver, and also then anticipate at each of your steps what are some of the potential hurdles or areas of constraint as you are transitioning into the next stage from a manufacturing process, analytical process, etc. So I think that those are things to consider. And that's where I see things shifting.
And Tim?
Mainly, I'm just going to echo what Kelly just said, to be honest. I think the two shifts you see at the moment is one in early stage pre-pivotal. Yeah, regulators will give you a lot of leeway, a lot of flexibility, get things right, change them, work out what your product is. But as soon as you say this study is pivotal, they want to see this process as a proper process. They want to make sure the product is aligning with that. They've got an eye to what they're going to be asked to approve after the study is going to be the same as what's gone through the clinical studies. There's a very big shift to be ready for and to plan for there when you go into a pivotal.
And Knut, from your experience, what would you say are the later focuses?
I mean, it's hard to follow these two because they mentioned everything that matters, right? It's absolutely right that the experience you will have with the agency in the phase 1 doesn't mean that it's going to be easy for pivotal. Pivotal is, as Tim mentioned, a very different beast, and be prepared for that. That's why I always say, you should consider your phase 1 trial almost like a pivotal, because any process changes, any changes in analytics, materials, it's not necessarily a given that this will be an easy change. Like, comparability is hard enough. If the agency decides that your comparability study doesn't show comparability, then you're set back for months. Same on the same note, potency assay. Don't think about potency assay when you're at the end of phase 2. You need it for the pivotal. You don't need it for phase 1, 2, but you want to have ideas what the potency assay looks like when you start the journey, because you want to have a very good conversation with the agency about the format of the potency assay. And as I mentioned earlier, if you look around, potency assay is an item that puts companies on CMC hold all the time. So I think that's a very critical piece to remember.
And how might this be reflected with stem cells? If we're looking at iPSCs, for example. Tim, I don't know if you'd like to take this one.
Yeah, I would actually. We do a lot of products that like from iPSCs. It's quite a fashion at the moment, and a lot of these people come to us, which I'm very glad about. But you can see how delicate these products and processes are. I'm really worried as to how consistent products can be when you change the differentiation process. And that can be even little things, apparently little things, reagents used in that process. These are quite delicate processes that may look fine academically, but convert them to commercialization. Well, we've seen developers who do manage to do that and do manage to get what look like robust products, but it's not a given. So you want to be very careful that you're not assuming too much before you embark on a phase 1-2 study, exactly as Knut said, with a product, and then you're going to change something and it's not going to be the same. I think with stem cell products, you should worry more than you should with any other product, and you should worry with the others as well.
Thank you. We are approaching the final portion of today's session. So if anybody in the audience does have any final questions they'd like to pose, please get them in. So as we return to the title of today's session, what would be your one piece of advice for developers looking to progress from early to later stage development of a gene-enabled cell therapy product? Knut, I don't know if you'd like to start.
Yeah, I definitely like it. It sounds like I'm repeating myself, but again, don't think early is different than late. Like, there is a big breaking point when you enter the pivotal trial. Obviously, you should have confidence in your asset that you get to a pivotal trial. So it is worth thinking very early on, not just the processing, but things we have seen as materials. People don't pay much attention to the material they use in manufacturing. Then when it gets closer to the pivotal, they realize that half the materials can't be purchased under the right quality, etc., GMP or USP. So design the process right from the start, like with a commercial eye, so to speak. And on the same note, the analytics are equally important. If you have a very sophisticated analytical assay for the quality control, that might work well when you have 1-2 patients a week, but think about when you have 100 patients a week and 100 products a week to release. Is that assay still working? So that will be my point here.
And Tim?
Very similar. Yeah, make sure the process is robust before you go too far.
Kelly, do you have any final advice?
So I think, although I agree with what Knut and Tim have both said. Again, looking at it from a regulatory lens. I know I've had numerous conversations with clients. I would say don't base your decisions on qualifications, validations, etc. Method, or not just method, but manufacturing process development on what is listed in the guidance document, because those regulatory guidances are evolving, and just because there isn't a guidance does not mean that when your reviewer looks at your submission, he's going to find it to be sufficient because you did the minimum that what you found in the guidance. A lot of times I will definitely have a client say, well, where did you find that in a guidance? Well, I don't have a guidance, but let's take that step back and let's plan. And let's look at this with some critical thinking of, again, look at that commercialization, just like Knut said, look at the end, right? The finish line in mind of, at the end of the day, we all want to be able to get this product on the market. And, you sometimes have to think outside the box of maybe what you want to hear, but you need to listen and do what will get you there and make you successful when the submission goes in.
Excellent, thank you. Well, that brings our session today to a close on that note. I think that's a really nice note to end on. So thank you to our panelists for joining us today. That's been a very insightful session and answer to our questions. Unfortunately, that is all we do have time for, so I apologise if we did not get a chance to address your questions. As always, though, you can access this webinar on demand via the Facilitate website and also via our network, along with many more of our previous webinars and other resources. You can also still register to join us in Miami in just a few weeks' time. You can find more information via the Facilitate website, and we are looking forward to seeing you there. That's all from me and from the panel. So until next time, thank you for joining us today and goodbye.
Thank you.
Thank you.